Hi, I'm Emily Mason, and I'm working in Dr. Anderson's lab working with Pseudomonas aeruginosa, which is a kind of bacteria. It's pretty common, but it causes problems in cystic fibrosis patients' lungs. It builds up and makes it hard for them to breathe. So I'm studying which kinds of the bacteria grow biofilms and which ones do not, so we can kind of get a good idea of how it grows biofilms and what genes are involved in that. So I'm screening our library of bacteria, which is in the fridge here, or the freezer actually. It's negative 80 degrees. I've got tons of these plates of bacteria that are in these bags, and I take them out and open the plates up, and using this tool, which we call a frogger, I transfer some of the bacteria from the stock into these little flimsy plates that I grow them up in an incubator overnight. First I'll put them in a solution we call LB, and then after incubating them for a day, I transfer them into M63, which is what these plates are. And then after incubating those for a day, I come over here and I stain them at the staining station. But this is what we stain them with. It's called crystal violet, and I wash them out, wash out the crystal violet, and then I look at them to see which ones grew biofilms, which would be indicated by a ring in the well. And from there I take note, and I have a file on the computer that tells me which wells have which genes knocked out. Right, so as a result of what I'm doing here this summer, I'm helping them to narrow down to figure out which genes are important for biofilm growth, so that they can study those genes more and kind of figure out how exactly they affect biofilm growth and what each gene's role is. So hopefully they'll be able to fight off the bacteria in cystic fibrosis patients better.
I'm Giovanni Biggerstaff, and my preceptors are Leland Plotkin and Matt Allen, and they're both PhDs. And my project is determining bisphosphonate concentration in the bone using in vitro assay of cell apoptosis. And so the first thing I'd like to get on is bisphosphonate drugs. These drugs are widely used in America alone by millions of patients, and what is really unique about them is that they work really well with osteoporosis. Scientists and doctors come along with this many issues about using these drugs, and one would be to how long does a patient need to be taking these bisphosphonate drugs in order to be effective, or how much is actually accumulated in the bone, because these bisphosphonates layer and layer upon the bone to help repair it. And also there's another issue of, do they need to be taking it for a year and take what they say a holiday or a break off the drug and then go back another year and be on the drug, or is it just a one-time deal? And there's also the issue of osteonecrosis of the jaw, or what they call necrotic jaw, which means the cells in your jaw are dying. And this picture right here will show that in the mouth here, you can see the bone coming out and the flesh dying. And this is pretty much the cells in your jaw dying, and it rots away and decays. And in some cases, you lose your whole jaw from that, and you get what they call a stage 3 of this necrotic jaw, which you can see right here, the spot, and what it does. And so there's that issue, and so using these drugs, they're very effective over time, but there's those issues where we don't know how much is in the body or how much to prescribe. So we go through all these experiments to figure out how strong the alendronate actually is, and how effective it is with the body, and how many cells it kills in your jaw. And throughout our experiments, we figured out that the alendronate is really effective, and it kills a lot of your cells in your skeleton. And so we use this method by MTT assay, which is basically a way to measure the growth and the depth of these cells, and that is by wavelengths. And we use that with a spectrometer, which we use as a reader. And so, in conclusion, I would say that by doing this experiment in this project, we are trying to figure out the way to stop necrotic jaw by the accumulation and determine how much to prescribe the user of these drugs.
Hi, my name is LaKenvia Leffert, and I work in the bioinformatics building along with Dr. Mita Pradhan and Dr. Matthew Polakal. This work focuses on global analysis of bibliometric data, including literature, to identify novel biomarkers for colorectal cancer. In our work, we are augmenting the experimental data of CRC with protein interaction data derived from literature to identify the biomarkers by analyzing all the pathways and networks at various stages of the disease CRC. Initially, we are analyzing the data without any clinical data, but once the clinical data is available, we use it to augment it to our analysis. This will benefit the society because observing the specific mRNA expression patterns will enable us to predict prognosis and response to selected treatments in CRC patients.
Hey, I'm Jacob. I work with Dr. Toon and Zach Berwick. We do mostly cardiovascular research on various animals. Earlier on in the program, we were doing a lot of in vivo stuff, a lot of surgeries. Very interesting. We did flow studies on the coronary arteries of several dogs. The protein we're looking for is called the intermediate conductance. The intermediate conductance calcium-activated potassium channel, which is a channel in the coronary arteries, and actually it's in a lot of other arteries too, but we're looking at it specifically in the coronary arteries. When it's activated, it causes the cell to, it causes the artery to dilate, to get larger, and it does this in response to things like regular, normal changes in blood flow in the artery, because that will change occasionally, just as a normal thing. So it sort of has to auto-regulate. So we think the channel deals mostly with auto-regulation, but we're not sure, A, because we don't know where it is, and that's what this immunohistochemistry is doing, and B, because the studies we've done so far on it are somewhat inconclusive, because the chemicals we have that can activate and deactivate it are a little non-specific, and they also work on other proteins as well. So we don't know if what it's doing is to that protein specifically or to other proteins. In terms of actual overall ways that it's helping humanity, I'm not sure. What we're looking into at this point is pure research. We are just trying to find out what this channel does. And once we found that out, who knows? Maybe it will help someone someday. It probably will. My philosophy on this is that all knowledge is good knowledge. Learning something new about something, even if it seems useless at the time, will probably be useful to someone at some point. Because it's all about, we're trying to figure out how this particular channel affects how the blood vessel dilates and contracts. If we can figure this out and it works in the heart, it could be used to, say, reduce cardiac disease, reduce heart attack, reduce problems with heart attacks, reduce diabetes, problems like that. We just don't know what its actual purpose will be in the real world eventually. We just know this is what we're looking for.
Hello, hi, I'm Janaea Hooks. I just graduated from Arsenal Technical High School. I work in the geology department of the School of Science in a volcanology lab. Basically what we're doing is we're studying the plumbing system of volcanoes, and we're trying to figure out what happens chemically before and after a volcanic eruption. Right now our location is in Cajon Pass in California. There was the Cajon Pass drill hole where we retrieved samples of intrusive rock, which means that the rock did not exit the volcano after eruption. It stayed in the volcano and then froze or solidified. and its crystalline structures are more defined. That's intrusive rock. So the intrusive rock was delivered to our lab. We then mounted them on slides, polished them, sliced them down to where they're at a translucent state and you can pass light through the crystalline structures. And what we were looking for is zircons. Zircons are pretty much the history book of the volcano. They record everything that goes on chemically within the environment of the melt as it moves throughout the magma system within the volcano. Right now, what I'm doing is looking at the chemical components and the elemental components within each zircon. Right now, the layers of the core that I'm working with are 13-3 and 17-9. 13-3 being the shallowest layer of the Cajon Pass drill hole, and 17-9 being the second shallowest.
But these are images of zircon crystals. And in these little green circles are where we have taken
The zircon crystals to an ion microprobe and blasted oxygen ions towards the specific sections within the zircon. And the oxygen ions break apart the covalent and ionic bonds within that zircon. And the atom particles get sucked into a spectrometer. And then they measure the components of each element, or they measure them in parts per million, so however much of one element is in that specific section of a zircon. The zircons grow in layers, so there's a core and then there's outer layers. And the core and each outer layer changes as the environment of the melt changes. So if there's an element that's introduced to the melt or the magma system, then the zircon records that within one of its layers. If there's an element that crystallizes and falls to the bottom of the magma system, then the zircon records that. What I do is I make graphs of the elements and the temperature of the melt at that time, because the zircon also records temperature. When you look at the layers of the melt, there's an equation you use, and depending on what elements are there at that time, you can calculate the temperature of the melt at that specific time. We compare measurements like these to the Madagascar standard. The Madagascar standard is a component that we use as our control, if you will, to compare all our measurements. The Madagascar standard is a series of zircons collected from Madagascar that were said to be at perfect constant in formation. The temperature of the zircons never really changed, nor did the amount of each element. Its elemental composition never really changed. So that's what we use as a comparison. Right now, as I said, I'm making graphs, and what we're trying to do is prove that there is significant variation in the elemental composition of the Cajon Pass drill hole compared to the Madagascar standard, meaning that the amount of elements does change drastically as the temperature drops and rises in a volcano, which then may lead to a more aptitude for eruption or less aptitude for eruption based on the amount of elemental changes going on within the melt.